For this homework video, we're going to talk about DNA mutations, so some mutations that occur during a DNA transcription. So the first type of mutation that we're going to talk about is a substitution mutation. So I've got a part of a DNA strand here, just the strand that we want, and so what we're going to do is we're going to talk about what would happen if some of these base pairs, some of these letters were changed, and what that would result in. So I've gone ahead and labeled them here just to make it a little bit easier for us. I've already labeled what protein each codes for. And so we're going to see what results, what changes in the proteins we see, yada yada, from different mutations here. So the way that this is going to work is I'm going to change something in the DNA, and then I want you to pause the video and try to hypothesize what you think would happen if that occurred. So the first change that we're going to make, we're going to substitute out that A, and we're going to put a C in there. So go ahead and pause the video and take a minute and try to figure out what that would result in. So hopefully you came up with some kind of hypothesis of what would happen if this occurred, if this A to C substitution mutation happened. So the way that we need to look at this and the way that we need to figure out what would occur is by looking at this little chart for the amino acid. So I know that it's kind of small, but I hope that you can read that on your screen. So let's look. So originally it was GTA. We know that on this amino acid table the T's are going to show up as U's, but let's go ahead and try to find that GUA. So look, we found it right here at the bottom. I'm not sure if you can see my mark there, my black mark. So GUA right above that black mark, and so it codes for valine. And so it's changed to GTC, so on the table GUC, so let's find it, it's actually right above. And so we see that it codes for the exact same amino acid, so there's no change in our amino acid sequence. So if our amino acid chain stays the same, what that also means is that our protein stays the same. So this mutation, what it's called is a silent mutation, where a single base pair is substituted, but it doesn't change our final protein. So there's no effect. And we see a lot of these silent mutations in our gene sequence, but they don't have any effect in the proteins. And so that's why you can kind of make sense of it. That's why it's called silent. Okay, so now it brought us back to our original DNA sequence. Let's talk about another substitution mutation. So this time it's going to be a little bit different. We're going to take out that base, that G there, and we're going to replace it with a T. So same as last time, pause the video, use that amino acid chart. I'll go ahead and put it out here for you. Try to figure out what results from that change. So just like the first one we did, now we're going to go ahead and find the original 3 nucleotide sequence, so TGG or UGG on the table, and find out what happens when we change that last G to a T. So on the table I can see it, oops, that was a little too low drawn there, so I can see it here, the UGG, and it codes for that TRP like we see. And so if we change that last G to a T, or it would be another U, then we can see it up here a little bit higher. It changes, if you see it on top of that second little blue line, it's kind of hard to draw on this, but it changes it to a UGU, which changes it to the CYS, the cysteine, a different amino acid. All right, so we're going to go ahead and change that amino acid there and put in the new one that it gets changed to because of that mutation. So this mutation isn't silent, it changes that single amino acid, so this can start to cause some issues. So this is what we call a missense mutation. So it's when an incorrect amino acid is formed that may cause a malfunction in our final protein. So the two that we have so far, throw them both up there, silent and missense. So those are both substitution mutations that do different things. All right, now we're going to talk about one last type of substitution mutation. So this time I'm going to take out this G and I'm going to substitute it, if I can get it in there right, for an A. So I'm going to throw this adenine in there. It's not perfectly centered there, but you still know it's there. So go ahead and pause the video, try to figure out what would happen in this case. So you should notice that in this case it changes from this TRP that it was originally, and it changes it to the stop codon. So as you know already, the stop codon is what's at the very end that signals the transcription process, the translation, to stop so that it doesn't create more amino acids. So what this causes is that now this amino acid is no longer coded for. It's now a stop codon. And we don't get any of the rest of these amino acids from the chain. So we only get 3 amino acids because of this, which can be a really big problem. So when this occurs, it's called a nonsense mutation, and it causes a shortening of the protein. And typically these proteins aren't even functional. And so this is the worst of these substitution mutations because it just cuts off a big chunk of that protein. So now we're going to talk real quick about an easy way, or a way that I came up with that is easy for me to remember these 3 types of mutations. Okay, so now I have up on the screen these 3 different types of substitution mutations that we have. So when I took this class, I really struggled to remember which one does what, because they're all kind of similar, and they all have these names that are kind of similar as well. And so, just like a lot of things in life, I tried to relate it to something I understood a little bit better, so I related it to dating. And some of you may like this analogy, some of you may think it's dumb, but either way it worked for me. So I thought of maybe a guy or a girl that was interested in somebody, but they were a little bit nervous to ask them on a date. So they approached their roommate and started asking some questions. They wanted to know, if this person was who they thought they were, if they were worth asking on a date. And so their roommate had only been living with them for about a month, so they knew a couple things about him, but they honestly probably weren't the best person to ask. And so they start asking questions, and the roommate tries their best, but some of the information they give is incorrect. And so you may think, you ask, well, what kind of music does this person listen to? And the roommate gives you a wrong answer. They said they like country music when they don't. So on the first date, you get in the car and you're playing country music, and turns out they don't actually like it. Well, to be honest, in the long run, I don't know that that's going to make the biggest difference or any difference at all in the relationship. So we'll go ahead and we'll call that and kind of compare that to our silent mutation, where it is a change, it's something that was incorrect, but to be honest, it really isn't going to make that big of a difference. So maybe next question, maybe something important to you is something like religion or politics. You want to make sure that this person you're interested in has same religious or political views that you do. And let's say that their roommate thinks that they do, and so they tell you, oh yeah, this person voted for so-and-so, and they go to this church. It turns out that that's wrong. So for a lot of people, that can end up causing a big issue in the long run because they got this misinformation. And so that's similar to our missense mutation. You get this misinformation, and maybe later on, it may cause a malfunction in the relationship. So then let's talk about this last one, a nonsense mutation. So this is, I don't know, maybe your roommate doesn't like you, she doesn't want the relationship to work, but she just gives you completely false information. Maybe she tells you that, I don't know, the person that you're interested in is dating somebody or already married, or something like that. One of those terminating things, addicted to drugs or a serial killer or something. And now you're like, oh, well, that kind of is a game breaker for me. It terminates the relationship. So that's similar to our nonsense mutation. It terminates the sequence. It stops amino acids. It gives that stop codon because the roommate told you something that was complete nonsense. And it turns out it wasn't even true. And so it's too bad because the relationship shouldn't have stopped, just like our coding sequence shouldn't have stopped translating, but it did. So that's kind of a fun and easy way to understand, at least for me, these things. I hope that it helps you. I hope no one was offended by any of the scenarios I came up with. But that was something that helped me throughout this class to understand these mutations. All right, now we're going to talk about transcription and translation, which is the process from going from DNA to the protein. So we're going to first start with transcription. So the following sequence here is for the hemoglobin gene, and this is a DNA sequence, so it's in the nucleus. DNA is always found in our nucleus. And so your job is to transcribe this DNA strand, the whole thing, from DNA to messenger RNA. So if you remember, this top strand, this 5' to 3' strand, is the coding strand, and the bottom is the anti-coding or template strand. So the coding strand is what we want, and so in order to get that, we make a copy of the opposite of the coding strand. So if we take this back to our dating analogy, there's the girl that we want to go on the date with, but we're a little nervous, and so we ask her roommate for information. And so what we want is this coding strand. So go ahead and do this first part, do this transcription from DNA to mRNA. So once you've transcribed your DNA, your messenger RNA will be single-stranded, and it should look like this. So this is still the unprocessed mRNA in the nucleus. So you see these bolded and unbolded parts. Bolded parts are the exons. Exons are the part that exit the nucleus. The introns are the parts that we snip out, that we don't end up wanting. So your next task is to go from this unprocessed mRNA to processed mRNA. So this is the preparation that's required in order to leave the nucleus. And so, like I mentioned earlier, you need to take out the introns, and then there's two other things. I'll let you think about what those are. Two things that you need to do to each end before it leaves the nucleus. So go ahead and take a minute and write out the processed mRNA. So this is what your processed mRNA looks like. The introns are all gone, so it's just exons left, ready to exit the nucleus. And then you add a 5' cap and then a poly A tail. So those are the two important things that are added to each end so that this mRNA strand isn't attacked and degraded as it tries to leave the nucleus and heads to the cytoplasm. So it's important that before it leaves that it has those two things on it. So anytime you have unprocessed mRNA, you find it in the nucleus. Anytime you have processed mRNA, you can also find that nucleus, but it won't be there for long. It's getting ready to leave the nucleus to head to the cytoplasm. So the next step from this RNA to our amino acid is called translation. So now I'm going to go ahead and give you this amino acid table here. So your job is to translate this processed mRNA into an amino acid chain. Pause the video and take a minute and go ahead and give me that amino acid chain. So this is the amino acid chain you should have gotten. Always remember that the stop codon, in this case UAG, doesn't actually code for an acid, it just codes to stop. So if you got this amino acid, if you understood this all, you should be good to go for all the homework and for the test.